Okay, let's take a look at exercise 25 on blood. So first of all, let's just break down blood and see what's in it. Blood is always broken down primarily first into what's called formed elements and plasma. So the formed elements are the cells of the blood. Everybody's heard of red, white blood cells and platelets, and everybody's heard of plasma, the watery part. Well, going back up here to the formed elements, the cells, they should make up around 45% of the total blood volume, and that's important because you have too little or too much of either one of these things, blood viscosity, delivery of certain materials and all that won't be what it's supposed to be for homeostasis. Those percents are important. But going back to the cells right here, red blood cells are also called erythrocytes. You hear that, erythro, in physiology and anatomy class? Think red blood cells. White blood cells are leukocytes. So you hear that leuko go with these. And platelets, thrombocytes. So that prefix thrombo will go with the platelets. So there's your cells. Now most everything in the blood is moved around by the watery part we call the plasma. Now the plasma is about 91 to 92% water. We see that just below right here. And should be about 55% of the total blood volume. There are countless numbers of things in that plasma. Water, ions, all sorts of dissolved substances, colloid proteins, clotting factors, antibodies, you name it. A lot of solutes found in that watery part of the blood. So looking a little closer at the plasma right there, again, water, electrolytes, nutrients, hormones, waste are all things you'll see in it, plus much more. As we said, about 91 to 92% of it ought to be water. And there's countless numbers of proteins. Now those proteins come in three big categories: albumins, globulins, and fibrinogen. The albumin's the biggest group here, produced by the liver, have a lot to do with blood viscosity, how thick it is and how it flows. It also works as a buffer. Most proteins have a negative charge that'll help to neutralize the positive hydrogen, keeping the blood and body from getting too acidic. Globulins right here. There's different types. You'll often hear about the antibodies. You'll see a lot more about these with adaptive immunity and lymphocytes and such. And then fibrinogen. These are clotting factors produced by the liver. A lot of things are needed for clotting. That's just some of them there. So let's go back to our cells. The red blood cells, again, are called erythrocytes. This is by far the most common cell type in your blood. Nothing else is even close to it. 1 little cubic millimeter, which is like a small dot, will have about 5 million red blood cells in it. So you've actually got trillions of reds inside your body. The shape of them is very important. They should have what's called a biconcave shape. In other words, they're going to be round like a donut, thick on the outside and very thin right in the middle. Won't be a hole like a donut, but think like a donut with a membrane still right there in the very center of it. This is going to greatly increase their flexibility. A lot of places red blood cells go, capillary, spleen, whatever, where they have to go through tight spaces. If they couldn't bend and flex, they wouldn't fit through there. Blood flow would stop, and that would always be bad right there. They believe the shape increases surface area just a little bit too. So reds, as far as their size, are about 7.5 microns. That's just a little bit smaller than a capillary, which are the smallest of your blood vessels. So they'll have to go through their single file. Reds only live about 110 to 120 days. Reason being, red blood cells lose their nucleus during development. They lose that nucleus to make more room for hemoglobin. That way they can carry more oxygen. But losing the nucleus means they lose the ability to repair themselves. And all that bending and flexing will wear away the cell membrane. And of course they'll rupture and lyse and all the contents spill out and the cell dies. Erythropoiesis refers to the production of red blood cells. And there's even a hormone from the kidneys called erythropoietin that tells the stem cells in the bone marrow when to make more red blood cells. And anytime your oxygen delivery is inadequate is when that would happen. Below it we see the platelets, also called thrombocytes. So that's your second most abundant formed element type in the blood. 250 to 400,000 per cubic millimeter with these, where again it was 5 million with the reds up above. Platelets have some of the materials needed for the clotting process. So clotting, coagulation be mentioned more in the lecture. Platelets also, remember, aren't even cells. There are these very big cells in the bone marrow called megakaryocytes, and the edges of those cells pinch off a little at a time, and that's where platelets come from. So they're just cell fragments. Then we've got the big category, our white blood cells called leukocytes. So look at the number of these. 5 to 9,000 per cubic millimeter. So look at, we went from millions to hundreds of thousands to thousands when we go through these formed element types. Now some of these right here will live a few hours. Some can live for months. Just depends on the white blood cell type. White blood cells also can move on their own. The reds and the platelets can't move on their own. They're like logs floating in the river. Wherever the current takes them, that's where they go. Now, white blood cells will be moved by the current of the plasma too. But they can move what's called amoeboid movement. If you ever saw an amoeba move, it's sort of like a blob that flows from one place to the next. White blood cells can do that. Again, the reds and the platelets can't. Now, there's two big categories of white blood cells. There are 3 grainy granulocytes, 2 non-grainy agranulocytes. So when they talk about granulocytes, 3 of the white blood cells, if you magnify them and stain them good enough, you can see little specks and grains, little cytoplasmic inclusions all throughout them. There's 2 agranulocyte white blood cells that don't have all those little specks and grains. So that's how they've been put in these 2 categories. So if we go back up here, look at our 3 grainy granulocyte white blood cell types. Now notice how these are also called polymorphonuclear white blood cells. Poly means many, morph means shape to their nucleus. They have many shapes to their nucleus. And by looking at their nucleus, that's primarily how you identify them. So with granulocytes, 3 of these white blood cell types here: neutrophil, eosinophil, basophil. Now, the neutrophils are by far the most common white blood cell you have. They live 10 to 12 hours. They die after one phagocytic event, so they're not going to live very long. And these have 3 to 5 lobes in their nucleus. Now, they've got one nucleus, but it's going to look like several. The lobes are like sections to one nucleus, and there's little tiny connections between them, even though often you can't see them. We mentioned these were the most common. Take your normal healthy person, 60 to 70% of all their white blood cells will just be neutrophils alone. The eosinophils here look very different from those above. Where the neutrophil have 3 to 5 lobes in a nucleus, these will have 2 lobes of equal size and shape. Often it looks about like a pair of sunglasses looking back at you. These are a lot rarer. These would be 2 to 4% of all the white blood cells that you have in your body. And we're going to look at the functions of these white blood cells further along too. Basophils are the rarest of your white blood cells. Now, they have 2 lobes a lot of the time, like eosinophils do, but they tend to have 2 unequal lobes. Another appearance their nucleus could have is to have an S shape to it. So different appearances to the basophil, a couple of possibilities. These make up 0.5 to 1% of the white blood cells in your average normal healthy person, so these are really rare right here. And they like to release chemicals like histamines. They act as vasodilators. They cause inflammation. A lot about these chemicals will be mentioned further along. They also produce and release heparin, a good anticoagulant. Keeps clots from forming when you don't want them to. So those are our 3 grainy white blood cells. Now here's our 2 non-grainy white blood cells, right? Don't have the little specks inside of them. So they tell you right here, the nucleus will take up most all the cell. When you look at a lymphocyte or monocyte, most of the time, the nucleus is just big and round, sort of like a fried egg, and the nucleus will take up just about the entire cell. You don't see that with the 3 above. They don't have the large, round nucleus. They've got lobes to it. So if you see lobes to the nucleus of a white blood cell, got to be one of those 3 up there. But if it doesn't have the lobes and it's just big and round, it's probably going to be one of these 2 right down here. So looking at our 2 non-grainy white blood cells, the first one mentioned here are lymphocytes. These are large, have a large unlobed nucleus. In other words, looks about like a fried egg, but where the yellow only takes up most of the space in a fried egg, the nucleus takes up practically all of the cell when you look inside that of a lymphocyte. These will generally make up 20 to 25% of the white blood cells, and they come in 2 major groups, B cells and T cells. So those are just 2 types of lymphocytes. The B cells, a lot of those will develop into what's called a B plasma cell, and that's where antibodies come from right there. These tend to mature in the liver and spleen, and again, they give antibody-mediated immunity. Very important when you get to immunity topics. Then you got the T cells right here. They provide cell-mediated immunity. These mature in the thymus gland, and they're more for destroying viral infected cells, tumors, and some bacteria, where the Bs are more about bacteria. But there's definitely an overlapping of their function. Again, that's a discussion for adaptive immunity. So again, two types of non-grainy, agranulocyte white blood cells. There's your lymphocytes we mentioned. There's also your monocytes. Now, monocytes are the biggest of all your white blood cells. Lymphocytes are the smallest. A lymphocyte is usually about the same size as a red blood cell, or maybe twice as big. Monocytes are usually about 4 times the size of a red blood cell. So these often look just alike, but their size is how you tell them apart. Look at one that's maybe about the same or just a little bigger than a red, or one that's several times bigger. That's usually how you tell those apart right there. Now, a monocyte might not just have that big round nucleus. It might also have a nucleus that looks like a horseshoe shape. They say kidney bean shape, same thing right there. And often 3 to 8% of your white blood cells are of the monocyte type. And something else we'll mention on these white blood cells, once a basophil has left the blood and entered a tissue, it's called a mast cell, M-A-S-T. Monocytes, once they leave the blood and enter tissues, are called macrophages. So monocytes go out into the tissues just like the basophils and other white blood cells do too. And then the monocyte enlarges and it's called a macrophage at that time. Everybody knows macro means large, things such as that. Now, one of the big reasons to look at your white blood cells, learn how to identify them, know what normal percent you should see with each, and then what disorders go with each one, is a very handy tool. So what can be done with white blood cells, a blood slide, and a microscope is something called a differential white blood cell count. So what you can do is this. Let's say you have a blood slide that's already stained. What you can do is scan across this slide. Usually about 400 power or more will give you the magnification you need. And every time you come to a white blood cell, what you can do is stop and identify it. Well, we just talked about how to identify these white blood cells right here. So every time you stop and see a white blood cell and identify it, what you can do is keep track of which ones you found and how many. Well, let's say you identify 100 different white blood cells on a slide. Well, after you've identified 100 of them, and if you kept count of which ones you found, there's our 5 different white blood cells, and how many you got of each, it's easy to get a percentage. So here's what you do. Let's say you did a differential white blood cell count. And let's say you looked at the percents that you found for each one of them. Well, if each one of these 5 white blood cells was within their normal range, well, then you can pretty much rule out these disorders that you see over here on the right. But let's say your neutrophil percent wasn't 60 to 70, but instead it came back something like 79%. Well, why would the body make more neutrophils? To destroy bacterial infections, because that's what they're good for, primarily there. You can also see elevated levels of these with appendicitis, but usually that's going to be due to a bacterial infection. If you see an unusually high percent of one or more of these white blood cell types, well, that means the body's making more of them. If you know what their functions are, well, that's what they would be used for. In other words, to help you get rid of whatever's going on inside of you. So again, let's say you did your differential white blood cell count. If you found that neutrophils were above 70%, somebody's probably got a bacterial infection. If your lymphocyte percent was above 25, probably a viral infection or antigen-antibody reactions. Could be even something like mononucleosis, whatever. If your monocytes were above 8%, then they probably got a chronic, an infection that's been going on for a long time. Tuberculosis is just one example of others. Your eosinophils, if they were above 4%, well, somebody could have allergies or maybe even parasites. Eosinophils block inflammatory chemicals. They keep them from spreading. That way they don't work too well in places where they shouldn't. But again, eosinophils also release chemicals that destroy parasites. Basophils, if they're above 1%, somebody probably has allergies going on because basophils, mast cells as they're also called, are where most of your allergy-causing chemicals come from. So those two have a strong correlation, and radiation exposure tends to increase their number and percent too. So again, if you actually counted up the number and got the percents you found on a slide for these cells right here, and then you look to see if any one or more of them is above their normal percent, well, then this tells you what's probably wrong with the person. Again, this is a very handy diagnostic tool. If you ever have blood work done, I guarantee you, some on that blood work, you'll see these 5 names right here. What percents were found for each? And if their percent was elevated, well, again, that tells a healthcare professional what's wrong with you right there to the right. So very handy little tool right there. Here's some questions, right? Very good to answer for review. Make sure you can draw a picture of every one of these little cell types right up here. There's pictures of them in your lab book, and we got descriptions up above. Look at those pictures. Think about the description and learn how to draw a picture of each. Know all this other information, like the normal numbers with like reds and platelets and white blood cells, the normal percents for the white blood cells, what disorders go with them, and so on down the line. Describe each white blood cell type, list the normal percentages for each one, things such as that right there. List common disorders that go with your different white blood cell types we see there, and so on down the line. Good little review questions for this information. So that's just a little quick review of our blood. We're going to look at that a whole lot more in other episodes and other videos.